Hi, good evening all. The fourth Thursday of the month has come around again, so we've got another brilliant talk lined up for you. As most of you know, we started off during lockdown doing weekly talks, but now that more live talks are happening, we've reduced this down to just one a month. And we continue to pull our speakers from far and wide. But again, as most of you know, our speakers are always up to date with the best evidence-based information in their field. Before I introduce tonight's speakers, just a few pieces of housekeeping. If you've missed any of our talks, you can find nearly all of them on our YouTube channel, although for various reasons there are a few that we can't give you a second chance to see. Also, a reminder you can support us via Patreon, Skeptics in the Pub online at Patreon, or you can go instead to donate us at sitp.online/donate. As always, QED is happening again this year. Yay! From the 19th to the 21st of October, and tickets are still on sale. Here at Skeptics in the Pub Online, just like last year, we're going to be setting up a hardship fund. QED is very reasonably priced given how much happens there, but it's still beyond the means of quite a few of us. So that's why we're setting up a hardship fund, and if you'd like to contribute, all contributions very gratefully received and going to a very good cause, please go to sitp.online/QED. We'll be opening it up for applications very soon as well, and that'll be at the same address. The day before Skepticamp, that is Friday the 18th of October, what am I talking about? The day before QED. Did I say Skepticamp? That's because the day before QED, Friday the 18th of October, we will be running Skepticamp again. That is a whole day of 10-minute talks. We always have a really great day, and the talks are always really varied and always fascinating. So we will be advertising soon for submissions, so keep your eyes peeled for that. On to tonight. As usual, our speakers will talk for about 45 minutes. We'll then be having a 15-minute break, and that'll be followed by a Q&A. If you've got any questions you'd like to ask of our speakers, and I'm guessing you'll have quite a lot, then please post them at sitp.online/ask. If you don't have any questions to ask, please go on and vote up the ones that you'd really like to hear the answers to. As always, a reminder, please be civil in the chat tonight. I'm sure you will. And now for the main event. Tonight, we've got three speakers who are going to talk to us about malaria. More specifically, I understand that they actually want to infect people with it intentionally and in order to study immunity, and they might even be asking for volunteers. So keep your eyes peeled for that one as well. I'm going to introduce you to Joe Salkeld, Katharina Grabowski, and Matteo Putra. Jo is a medical doctor with an interest in infectious disease. She's worked on the COVID vaccine and is currently at Oxford University working on human stage malaria and researching vaccines. Matteo and Katharina are both at the University of Edinburgh in the Department of Biochemistry, and they're working with the Oxford Vaccine Group. Matteo is a PhD candidate and Katharina a research assistant. And tonight they're going to talk to you about malaria.
Thanks for this lovely introduction, Cleo. As mentioned, Matteo and I are researchers at the University of Edinburgh, and we've been working in very close collaboration with the University of Oxford for years, and I'm happy to share a bit more about what we do. So Matteo will take it away now with a bit of the background to malaria and why we're researching it.
Good evening, everyone. My name is Matteo. I am, again, a PhD student or candidate at the University of Edinburgh. And we do exciting research in malaria immunology. So before we start, I think that I might know that everyone might know what malaria is, but I feel like it's good to have some refresher on what this nasty disease is. So let's start with some morbid facts. There's a big world map there, and in fact, half of the world's population is actually at risk for malaria. And you can see where all the red is, because most of the burden is actually in sub-Saharan Africa, even though some other cases also happen in South America and Southeast Asia. And unfortunately, most deaths, actually more than 500,000 each year, occurs in children less than 5 years old. Not a great thing at all, is it? And malaria itself is a really complicated complex disease because it can range from asymptomatic cases to deadly symptoms. For example, liver damage, brain infections, also we call it cerebral malaria, fever, and also anemia. And what is actually malaria? Again, it's an infectious disease that's caused by Plasmodium parasites. So these are really, really small parasites that you need to use microscopes to see. And they're actually transmitted by the bites of female Anopheline mosquitoes. So these mosquitoes generally thrive in tropical climates, for example, in sub-Saharan Africa, but also in South America and Southeast Asia, as I mentioned earlier. And so imagine yourself, you may be are doing a trip, you're enjoying your trip, and then a mosquito lands on your skin. And then it bites you, and what it actually does, if you're unlucky enough that the mosquito is infected by malaria, it will then inject the parasites into your bloodstream, and then the parasites will then travel to your liver. And your liver, they would actually reside there for at least around a week, and they would divide thousands of times. And after that, they would then go out from their liver, get into the bloodstream, where then they would invade red blood cells. And inside those red blood cells, they would then divide again. And in fact, each red blood cell can create 10 new parasites every 2 days. So after dividing, they would then burst outside of these red blood cells. And that's why we get anemia when we get malaria. So this can continue for weeks and months. And if you can't access the medication, then you can be really, really unwell. And if you're unfortunate enough, then the parasite can continue its life cycle because the mosquito can then bite you, get some of your blood, get some of the parasites, and continue the life cycle again. So malaria is a really nasty disease, but also with a really, really complex life cycle, which Joe will talk more about later. Why is malaria such a big issue? So again, there's a morbid graph on the right side over here, because that's yearly deaths. And we can see that from the early 2000s to 2015, there's a good reduction in the number of deaths. But for the last decade or so, the progress has been slowing down against malaria. This is due to multiple factors, for example, the COVID pandemic, but also the issues such as resistance against antimalarials and also insecticide resistance in the mosquitoes, which is why it's really important for us to do malaria research. And there are also other morbid reasons. For example, if you get a severe malaria, for example, if the parasite goes inside your brain and causes cerebral malaria, even if you survive, you can have long-term consequences, for example, cognitive issues. And in fact, malaria can also have a huge economical impact. Some studies estimate that there can be a reduction of 5 to 6% of the country's GDP affected by malaria. And last but not least, most of the people that gets affected the worst are the children, and we really don't want that, do we? How do we tackle this disease? So we can break that down to 3 main ways. The first one, the most obvious one, is to prevent the mosquitoes from biting. So you can either spray the insecticides to kill the mosquitoes, or you sleep under bed nets that are treated or untreated by drugs. Or you can try to kill the parasites inside the blood using drugs, for example, Malarone. Or more recently, scientists have been trying to develop a vaccine to try to kill the parasites inside the liver before they can actually go into the blood using recently licensed vaccines. But unfortunately, these strategies don't always work, which is why it is imperative, really important for us to understand malaria better. And we have the right model, the right way to do that through controlled human malaria infections. As teased earlier, we actually deliberately, in a controlled manner, infect volunteers, I would say lucky volunteers, in clinical settings for us to be able to understand in a medical research term what happens when someone gets infected with malaria. So where did this crazy idea actually come from? So let's travel back in time to the early 1900s, where there are 3 main diseases that concerned humanity at that point: tuberculosis, syphilis, and also malaria. So there was this crazy Austrian psychiatrist forgive me if I pronounce this wrongly, Dr. Julius Wagner-Jauregg. And he was doing some studies on neurosyphilis. So neurosyphilis, or severe manifestation of syphilis, is a sexually transmitted infection caused by the bacteria Treponema pallidum. And if this disease gets severe enough and infects the brain, then it causes neurosyphilis. It's really, really nasty form. So this crazy psychiatrist had the idea to okay, let's induce fever by infecting these people with a species of Plasmodium called Plasmodium vivax. And it somehow worked. Some or most people actually recovered from neurosyphilis. And several important note, this is a crazy thing that we don't do anymore, because this was prior to the clinical utilization of penicillin in the 1940s. And we don't really want to get severe fever from malaria, do we? Moving forward, closer to the 2000s, 1980s, CHMI was then used for vaccine development because they realized that, CHMI is actually, or controlled human malaria infection, actually provides a really safe and well-controlled framework to evaluate the efficacy of vaccines. Again, Joe will definitely talk more about this later. And also, this will allow us to evaluate drugs, but also study the immune response, which Katharina will talk more about later, and also diagnostic tools for malaria. Controlled human malaria infections are now currently being done all across the world, in Australia, in the USA, and also in the UK. And there are two main ways where we can inoculate or infect the participants with the parasites. On the left, done in the USA, you can infect the participants by letting the mosquitoes bite them. So they would inject these form of parasites called sporozoites, and the parasites would then happily swim inside them. And we monitor them really closely. On the right, we can then inject them with parasitized red blood cells. So red blood cells that have parasites inside them. So this is my last slide. Hopefully I haven't bored you so far. But at the University of Oxford, they have been doing CHMI for at least 20 years. And they have done this for more than 600 participants. So it's a really, really established way to do research down in Oxford. And they have produced so many quality research from that. And no severe malaria at all. But that's all from me. I think I would hand it over to Jo for her to be able to talk about vaccine developments using CHMI.
Yes, so my name is Jo Salkeld. I'm a doctor at the University of Oxford. I work in the Department of Biochemistry with the Draper Group. And our bread and butter is very much malaria vaccine clinical trials. And some of these also involved controlled human malaria infection studies, or challenge studies, as they're more simply called. So I'm going to talk a bit about how vaccines can target malaria parasites, how they're tested, and how malaria challenge studies fit in with this testing. How does a malaria challenge study work in practice, and what is the volunteer perspective? And how have these studies contributed to the malaria vaccine development for the vaccines that we have? And then a bit about what the future holds. So how can vaccines target malaria parasites? So as Matteo has said, the malaria parasite life cycle is very complex. So it involves stages in both the human and the mosquito. And this has made vaccine development very difficult. But it also means that there are different stages of the life cycle that we can target with different vaccines. So the two most advanced malaria vaccines that have now been recommended by the World Health Organization are both what we call pre-erythrocytic vaccines. So these are vaccines that target the very early stage of infection. So just after the infected mosquito has bitten a human and injected the malaria parasites into the body, and before they reach the liver. The stage that our group works on in Oxford is the blood stage of infection. So this is after the parasite has been released from the liver and when it's reproducing within the human red blood cells. Now, this is the stage of the infection that actually causes the symptoms and the complications of malaria. And it's also the stage of infection which people can develop natural immunity to malaria over multiple malaria infections. Then finally, we have what are known as transmission blocking vaccines. Now, these are interesting, so slightly different in terms of vaccines. They don't protect the person who's been vaccinated. There's no effect on the malaria infection in the body. What it does do is stop the malaria parasite reproducing within the mosquito. So it means they can't transmit malaria onto anyone else. So how do we test these vaccines? Now, I think this is something, vaccine development that people have become a lot more familiar with over the past 5 years with COVID vaccines. And you'll have seen in COVID that vaccines were developed, gone through all the phases of testing in less than a year. An amazing achievement, but very much not normal. So the first malaria vaccine that was developed, so RTSS, took about 20 years to go through all these phases of testing. And the vaccine testing, it generally starts in small numbers of healthy adult volunteers in Western countries which don't have malaria infections, before then moving on to small numbers of healthy adults and children living in countries where there is malaria transmission. Now, both these phase 1 studies, or what we call phase 1 studies, are only in tens of volunteers in each study. And they look at the safety of the vaccine, they look at the immune response to the vaccine, but they don't give us any idea as to how effective the vaccine is in real life. So we can then move on to phase 2 and phase 3 studies, which also take place in countries with malaria transmission and take place in hundreds in the case of phase 2b studies, or thousands in the case of phase 3 studies of children living in these malaria-affected areas. And at this point, we're getting an idea of how effective the vaccine is in real life, as well as more information on its safety and the immune response. So how do malaria challenge studies fit into this? So they come in midway through the development process. So after you've infected tens of volunteers and you have an idea about safety and the immune response, and now you're trying to work out whether you take the vaccine to these larger scale studies that involve hundreds of thousands of people in African countries. Now, these studies, they're slow to set up, they're complicated, they're expensive, and they're slow to give you results as well. So you really need to be reasonably sure that it's going to work before you commit to something like that. So challenge trials fit in here, and they help us make that decision. So again, these have traditionally been done in Europe or North America, in countries where there are no malaria infections, and they're done in small numbers of healthy adults. They allow us to infect both a group of volunteers who've been given a malaria vaccine, as well as a group of volunteers who haven't had any vaccine. And they allow us to compare between the two groups whether they develop a malaria infection and how fast the parasite grows in the blood. So it gives us an idea of whether the vaccine will be effective in real life. Now, malaria is particularly well suited to these sort of challenge studies. There's a short time period between infection and the development of symptoms. We have well-established diagnostic tests and effective treatment. And there's also no risk of long-term health problems or transmission of malaria infection to others following the treatment. So as Matteo has mentioned, since the 1980s, there have been a lot of these studies. Over 2,500 volunteers have been infected worldwide with falciparum malaria, which is the type of malaria that causes most of the cases and deaths that we see in the world. And here in Oxford, almost 600 volunteers have been infected in these studies. And it's important to say that there have been no cases of severe or life-threatening malaria in any of these studies. So how does the study work? Well, first of all, we need to find the right volunteers. Very important to get the right sort of volunteers for these very intensive studies. So the first thing we do is want to make sure that people understand what they're signing up for. We give people an information sheet to read through. It's 20 or 30 pages, so it's a lot to take in. So we get them to complete a questionnaire. These are not difficult questions. There's no trick questions. It's things like, what disease might you develop if you take part in this study? How will we give you this disease? Will the treatment that we give you work? If they answer all of that correctly, we can go ahead in terms of the screening of the study, and we have very strict medical requirements because we're aiming to enroll a very healthy population. So once we have our volunteers, they're ready and willing, we need to give them malaria. And as Matteo mentioned, there are two ways that we can do this. The first is via mosquito bites, so the same way that you'd get malaria in the wild. Although in this case, we use lab-reared mosquitoes, which we know are infected with a specific lab-adapted strain of malaria. The other way that we can infect people is via injection of infected red blood cells. And this is what we usually do in Oxford when we're working on the blood stage vaccine, because this is the stage of the parasite life cycle that we're targeting. It allows us to give all of our volunteers the same dose of parasites and also a lower dose than if they'd had a mosquito bite. And so it allows us to follow them up for longer and monitor the parasite growth for longer before we treat them. So once we've given people malaria, it's very intensive follow-up because safety is paramount. It's not an inpatient study, so people go home every day, but we do have at least minimum daily contact with them. This is either a phone call initially, or once we think that they might start to develop symptoms or have a detectable amount of parasites in the blood, then we bring them in once or even twice a day, and we check their parasite count at every visit. So our requirement for them is that they stay in the Oxford area so that they can come in quickly if needed, and we also have a study doctor available on call 24 hours a day. Now, this is a potentially fatal infection if untreated, so we need to be sure that we can get hold of people. We get them to give us their next of kin details, so we'll contact them initially if they don't turn up for a visit, but we do warn volunteers that their next of kin doesn't know where they are, or we can potentially get the police and national media involved in looking for them. So symptoms that people experience in these kind of studies, they're very variable. We'll have a few people who don't get any symptoms before we treat them, but for most people, if it's the first time that they're being infected with malaria, they will experience symptoms. And most commonly, these are things like headache, tiredness, feverishness, sweats, muscle aches. So it's a flu-like illness, and some people may feel pretty grotty for a couple of days. But as I said, there's never been any cases of severe or life-threatening malaria, and we treat people at a very early stage of infection, so far earlier than you'd ever be diagnosed with malaria if you got a malaria infection in the wild. We also infect people with parasites that we know are sensitive to the standard malaria treatment, so we give them tablets and we can be confident that it will clear the infection. And we do also monitor the parasite count in the blood as a fail-safe, just to make sure that there's no parasites left. If their volunteers do not meet the criteria for treatment, they get treated at day 21 anyway, even if there's no evidence of malaria parasites in their blood, just to be doubly sure everything's safe, everything's treated. So, I mean, you've heard from me, my perspective about what it's like, but what do our volunteers think about it? So I've just got a 2-minute video that I'm going to share with a couple of volunteers talking about their experience.
My name is Kate. I'm a content project manager.
So my name's James. I'm 28 years old. 2 years ago, I joined one of the malaria challenge trials here in Oxford.
Yes, so the malaria challenge study, it involved being injected with malaria parasite-infused blood. After having been injected with blood, it involved coming for daily visits, so over a period of 2 weeks. And they were just to take a blood sample and blood pressure, etc., vital signs. So it was probably like 5-10 minute visits, not too taxing.
So the actual challenge itself, pretty quick, no big dramas, nice and easy. I was thinking, malaria, horrible tropical disease, going to be stuck in bed for weeks. But no, the actual symptoms themselves weren't anywhere near as bad as I was expecting.
The study team wanted to make sure that I am safe, and the minute that the parasite count is high, the treatment would kick in and get started. So I knew that even if my symptoms were severe, I was in good care.
So the whole team was super supportive. I was really well looked after throughout the whole thing, so I didn't have any concerns. So the various strains of malaria affect a few 100 million people every single year. In the global south, it kills tens of thousands of people every single year. So if it's possible that the work here at Oxford can help create a vaccine to prevent that from happening, that's then going to save thousands and thousands of people from suffering that doesn't have to happen.
If I, as a healthy person, could be part of a potential vaccine that could help many, many lives worldwide, why not do that?
Okay, so that was the perspective from a couple of our volunteers. And how have malaria challenge studies helped malaria vaccine development so far? So both of the recommended malaria vaccines have been tested via challenge studies. So as I mentioned, these are both pre-erythrocytic vaccines. They target that initial stage of infection. So they're trying to prevent the parasite getting into the liver. For this sort of vaccine, once the parasite is released from the liver, once it's detected in the blood, it's game over. We know the vaccine hadn't worked. So it's a simple sort of yes-no question. The type of vaccines that I work on, so blood stage vaccines, are a little bit more complicated because we're not trying to prevent the infection of the blood. We're hoping that we may be able to control the parasites in the blood. So it's not necessarily game over if we detect the parasites in the blood. Instead, we monitor the parasite growth, see how fast it grows. If we have an effective vaccine, we expect it to grow more slowly. And if we have a really effective vaccine, we may even see the parasite growing and then the parasite count going down as the body is able to clear the parasite from the bloodstream. So the two malaria vaccines, which have now been recommended by the World Health Organization, both target the same part of the malaria parasite, so a particular protein on the surface of the parasite just after it enters the body. The first vaccine was called RTSS, and this was developed and tested over about 20 years. They did challenge studies of this vaccine in the US, where it showed approximately 30 to 50% protection against malaria. Then going on to test this in the field, it showed approximately 36% protection against malaria over 4 years in young children. And then there have actually been further studies that have shown it has a higher effectiveness when it's combined with seasonal malaria chemoprophylaxis. So this is when all young children in an area of high malaria transmission get regular anti-malarial treatment over the malaria season, whether or not they have any symptoms of malaria. And so this was recommended by the World Health Organization in 2021 for use in areas of moderate or high malaria transmission. The next malaria vaccine was R21, so that was developed here at Oxford and tested in challenge studies in Oxford as well, where it showed up to 82% protection against malaria in these challenge studies. Going on to the field, it shows 75% protection against malaria over 12 months in young children and was recommended by the World Health Organization in 2023. So we've got our first malaria vaccines. This is a hugely exciting time for the malaria vaccine field, but we're not there yet. Both vaccines will need annual boosting to maintain these high levels of effectiveness. So what can we do to make better vaccines? And this is where we're coming in, and we're looking at the blood stage of infection. Our vaccine, our most advanced vaccine, is called RH5.1, and this targets a particular protein that's on the surface of the malaria parasite during this blood stage of infection. And this particular protein is appealing because there's not much variation in this protein and the different strains of malaria. So we think it can be effective across various strains that are in circulation around the world. So we tested this in a challenge study in Oxford. And again, this isn't a simple yes-no question. Does it prevent malaria or not? We're looking at the rate of parasite growth in the blood. And in this challenge study, it showed a 20% reduction in that growth rate. And this was the first time that a blood stage vaccine had ever shown this. So that was exciting for us and spurred us on to develop the vaccine further and to test it in the field. Now, the next numbers are sort of hot off the press. So we've just got these results in. We are doing a phase 2b study in Burkina Faso. So if you remember back to the slides about the various stages of infection, this is in hundreds of children living in an area of malaria transmission in Burkina Faso. And the vaccine has now shown 55% protection against malaria over 6 months in these young children. Now, this is still early. The study is still ongoing. The 6-month figures were from March, so our next results that we'll get will be at 12 months in September. So where are we going from here? So to start with the positive news, so RTSS, the first malaria vaccine, has been used in a pilot study in Ghana, Kenya and Malawi since 2019. It's been given to tens of thousands of children. And we're starting to get some really positive numbers coming out of there. So they've seen a 22% reduction in hospitalization for severe malaria and a 13% reduction in all-cause mortality. So this isn't just malaria-related deaths, this is a 13% reduction in deaths from any cause. We're also starting to see the rollout of malaria vaccines as part of the routine childhood vaccination schedule. So there's at least 30 countries in Africa that have put their name down as keen to receive these vaccines. And Cameroon was the first country to introduce the vaccine as part of its routine schedule in January 2024. And we have a nice photo there from BBC News showing the first child to receive the vaccine in Cameroon. But how can we do better? You know, these vaccines, they still have limited effectiveness and there's a limited duration to this effectiveness in protecting against malaria. So we hope that our blood stage vaccines may be able to give long-lasting protection against severe malaria by controlling the parasite growth. And so that's something that we're continuing to look at in our study in Burkina Faso as it continues. And we've got a challenge study here in Oxford where we wanted to know if we give people repeat malaria challenge studies, can this give us a proxy to measure this effect? And this is a study called Bio4. We also want to know whether we can make a more effective version of this vaccine. So if we take this malaria parasite protein, and rather than using the whole protein to make the vaccine, we just use that part of the protein that triggers the largest immune response. Does that give us a better vaccine? So we've also got a study in Oxford looking at that, and that's called Bio1. And we also want to know what happens if we combine vaccines, because we've got the vaccines against the different stages of the life cycle. If we take one of these early stage vaccines and we combine it with one of our blood stage vaccines, do they work together to make an even more effective vaccine? Or do they actually interfere with each other and make each other worse? So we don't have the answer to that yet, but we've got our first multi-stage vaccine trial coming up in Burkina Faso next year. So if you want to know more about our trials, then please scan the QR code on the screen and it will take you to a page about our studies. And if you're 18 to 45 and living in the Oxfordshire area or know anyone who is, then we're always looking for volunteers. So in conclusion, malaria has not gone away. It's still a major global health problem. Children are still dying, and 1 child dies every minute from malaria. The first vaccines are now available, which is fantastic, but these vaccines can be improved, and challenge trials can help us make these better vaccines. So the work in Oxford is continuing. So thank you, and I'll hand over to Katharina.
Thanks, Jo. So like I mentioned before, my name is Katharina, and I work in Edinburgh in the field of malaria immunology as a research assistant. So now that we've heard about controlled human malaria infections and why they're so relevant for vaccine development, I'd like to talk to you a bit more about why we biomedical researchers are also very interested in this technique. So how does immunology play into malaria? So immunology is the study of the immune system, in our case, the human's immune system. And we want to understand how the immune cells that build up the body's defense against infections and infectious diseases work, especially in the context of malaria. Now, it is actually a difficult job to do because malaria is such a complex disease based on the fact that the Plasmodium parasite is a very complex parasite. So Joe and Matteo have mentioned the elaborate life cycle that the parasite goes through. And other aspects that illustrate its complexity are its large genome. So this parasite has many, many more genes compared to bacteria or viruses, for example. So 1,000 times more than a bacterium, for instance. And at the same time, the malaria parasites have co-evolved with us humans for a very long time, meaning they've gotten very good at evading our body strategies to get rid of them. And so immune mechanisms that would otherwise be very effective against pathogens like bacteria or viruses do not work in the context of malaria because the parasite is just too clever for that. So we want to understand still, how do people develop immunity? Because although the death toll is still huge from malaria, and we want to prevent any death that we can, at the same time, most people that actually in the world across all age groups are exposed to malaria survive this. Because there are many areas, as Matteo and Joe have mentioned, where malaria is quite prevalent. So how do people manage? How can it be that one pathogen, and sometimes at the same dose, if you will, out there in the real world can affect somebody so badly that they might die, or somebody else might be completely asymptomatic? So at this point, I think it's important, but also quite interesting to consider that there are different definitions of the term immunity. On one hand, there is immunity from disease, which means that your body knows how to employ its immune system so that you do not get a severe form of an infection. On the other hand, there's the immunity from parasite, which means that your body knows how to clear the parasite using its immune system. And one way to illustrate immunity from disease, for instance, is COVID. If you all remember, the vaccine that was employed there was especially helpful in preventing severe disease. So they really help people to build up an immunity towards disease. So although you might get the vaccine, you might still get COVID, but chances are much more low that you would die from it. Whereas there are other vaccines who aim for your body to eliminate a pathogen. So this type of distinction is quite relevant for malaria in particular. And although we do not fully understand how exactly our immune system manages to either cope well or not so well with this malaria parasite, we're really keen in understanding this better, and that's where our research comes in. So as I've mentioned, although we've been interested in this disease for decades, there's still very little understanding how the immune system actually manages to successfully deal with malaria. But there are quite a few epidemiological conclusions that we can draw. So epidemiological data, meaning data from people in areas where malaria is quite prevalent, having their cases, how severe their cases are, and whether they die from it or not, and what age they are, can give us a lot of information as to how, in a broad sense, immunity can work. And for decades, we have known that, as mentioned before, children are the most vulnerable. Most deaths occur in children. And this is actually not a coincidence, but is linked to the fact that in those areas, your first infections in life will happen in your childhood. So epidemiological data has also shown, linked to this, that the risk of severe malaria, which of course is then related to your chance of dying from malaria, decreases quite significantly with every infection that you experience in your life, as you can see on the graph in the graph on the right here. So the good news is that we know that immunity to severe malaria, so immunity against disease, develops quite quickly. And even if children are reinfected with the same number of parasites, they're able to deal with it better the next time. So how does that work? Pathological data also has shown us that severe cases of malaria are mostly linked with an extreme immune response. Again, this is kind of similar to what we've observed in COVID. Research into COVID has shown that people who have severe COVID, often the issue is actually that their immune system is reacting very extremely, and thus that exacerbates extreme consequences for their health. So what that means from our epidemiological data is that the immune system is acting over initially in your first malaria infection or in your first few malaria infections, but then gradually your immune system learns to adjust this response into a more safe manner. So we want to understand this better, how it works, how the immune system manages to adapt this way. Now we have chosen to study this with controlled human malaria infections. So why would we want to choose controlled human malaria infections as opposed to other approaches? In biomedical research, for instance, animal models like mouse models are quite common, but we choose not to use them because although there is malaria in mice, immunologically it's quite different from humans. And although we are at the end of the day interested in understanding the immune response in children in areas where malaria is quite prevalent, it is very difficult to actually do immunological research in those affected areas because, first of all, we're dealing with children who are quite vulnerable. So ethically and logistically, that's quite difficult because, of course, we want to ensure that they're protected the best and any infection that they do have is cleared as quickly as possible so that their safety is guaranteed. And another logistical reason is that we have no way of knowing when a child who lives in an area where malaria is very prevalent, when they get the first immune infection. So due to those reasons, we have opted to use a CHMI on consenting adults here in Oxford. And we have already done this a couple of times in close collaboration with Joe and her colleagues. And so far, our studies have shown that indeed the immune response that we can closely observe in a CHMI because of the controlled setting, because of the resources it offers us, so immune response mimics quite well what we observe in Africa, for instance, meaning that upon repeated infection, the immune response changes towards a trend that seems to be less extreme and associated with fewer risks of negative consequences for one's health. So our previous CHMI trials also double-checked whether this might be that because participants are better at killing parasites with their own immune system. But as you can see in this plot over here, the growth curve of parasites looks the same no matter if it's the first, second, or third time that somebody is exposed to malaria, meaning that participants do not get better at killing parasites at all, and yet their immune system is changed to a way that models a healthier outcome if this were to happen out in the field. So those are quite interesting findings that we have made so far. And going forward in future trials, we want to now properly understand how does the immune system manage to adapt. Oh, the cat wants to say hello. Come here. Cat run away. So how does the immune system manage to adapt? And does that come with any effect on the response to other pathogens? What are the pros and cons of this adaptation? So these are all questions that we want to understand in our research team And together with Jo and her colleagues, we're setting up this clinical trial, Bio04, to look deeper into those questions. So thank you very much for your attention. We hope we were able to spark your curiosity. Oh yeah, last slide. Just to summarize, we want to understand the underlying immunology in order to better inform vaccine development, in order to be able to understand how come some vaccines work better than others, in order to inform better in what capacity to employ them, but also optimize and inform the use of anti-malarial drugs and other prevention strategies out there, so as to be able to best harness the naturally developed immunity that people get upon repeated malaria exposure. Now, that is all I wanted to say. So if you do have any questions, pop them in the platform provided, and we'll be happy to hear what your thoughts are. Thank you.
Well, thank you very much indeed. That you've left me feeling really hopeful, and that you're doing something really important. And it's always lovely to hear about these things going on that are positive and heading everything in a good way. Well, that was very clever, but you all know what I mean. And I imagine quite a few of you have had similar responses to me. So if you have, then, and if you're in striking distance at all at Oxford, then I do hope you were snapping at the QR code. Even if I struggled to fully get on board with Matteo's idea about you being lucky to be infected with it. It's a good thing to do. We are going to go into our break shortly. Just a reminder that next talk will be on the 27th of June. That's the last Thursday in June. That after the Q&A, we will be opening the lock-ins razor. Don't forget to go to sitp.online/pub. And during the break, please post your questions for our speakers, and do head along to sitp/ask to upvote the questions that you'd really like here answered. So we will be back in 15 minutes at about 8:05. We'll see you then. Hi everyone, welcome back. I hope you're all suitably refreshed and ready for the Q&A. I've had a look at some of the questions, and it's going to be a really interesting Q&A, and we're going to learn even more than we already have. Except for the first question, maybe, because that is our regular, and this time it comes from the SIPPO Collective. And we ask the pet question. Show us your furry friends. Anyone able to go about that one?
Here's my cat staring at everyone.
A very regal cat.
Maybe he will come a bit closer in a second.
Oh.
Hello.
That's the only pet deigning to be with us this evening, is it?
It seems so.
Unfortunately, yes.
Right. We'll move on to this is becoming a bit of a trope, isn't it? Igor is our always asks excellent questions, and lots of them. So next question is from Igor, who asks us, this is following on, you were saying how we all learned a lot from COVID, and you can feel how that's influenced this question. Is malaria parasite one of those constantly mutating evils? Is it static and stable?
I'll just start it off. It surely doesn't evolve as quickly in a way that affects our research as COVID did, I'd say. However, it does evolve, as we see in the rise of anti-malarial resistances. So certain medicines, certain malaria strains are getting resistant to, meaning that those medicines don't work as well anymore in certain areas, which is certainly a big concern that many people have. But it's been a pathogen that's been around for absolute ages. So in that sense, it's not as fast moving if we think back to COVID and all the variants that required us to get boosters. So it's probably other reasons that we need to get boosted with malaria vaccines if we're to get them.
I'm not sure anyone asked a question, but do you mind if I ask a follow-up to that? Because we all know from COVID that the reason we have to have boosters is because it keeps changing. You hinted that there's a different reason for malaria?
So, I mean, malaria vaccines the ones that we have so far only give fairly limited duration of protection. So in all the trials, they've been giving yearly boosters. So it's exactly the same vaccine. It's not because it's mutated. It's just the immune response drops off quite quickly. And I mean, that's one of our research questions. Can we make a vaccine which lasts longer? Because yearly boosters is not particularly practical, especially when you're talking about countries in sub-Saharan Africa that don't necessarily have an awful lot of health infrastructure. These countries tend to have well-established childhood vaccination programs, but they give the vaccines at certain time points. So it's very, very difficult to come and say, actually, we need to give these extra vaccines every year. So it's something we're still working on.
Right, thanks. The next question comes from Serda, and he refers back to COVID again. He says, COVID-19 must have affected malaria research, funding-wise, and the general focus. Did malaria research benefit at all from the pandemic?
Katharina, do you want to answer that?
Yeah, I do have an idea about that. So, of course, in terms of funding, some funding might have been sidetracked to COVID, but I think a positive, if you can call it that, effect that COVID, and especially the success of COVID research and COVID vaccine have had on malaria research, is that it kind of opened some people's minds up in terms of the immunological approaches that we could aim for when developing novel strategies, when investigating what immune responses actually happen in malaria. And what I mentioned during the talk, differentiation between immunity against disease and immunity against parasites or a pathogen in general, that kind of plays part into that. So since the COVID vaccine was so effective, but it was designed to prevent severe disease rather than people getting COVID in the first place, I think people in the malaria field are now more open to consider a similar type of approach in malaria as well. So can we develop strategies that perhaps don't lead to you getting rid of the pathogen altogether in your body, but for you to deal with it better so that you don't get severely sick?
Yeah. Just to add to that question, I guess the good thing about, what the pandemic has been doing to us is that I feel like a lot of people now care more about what infectious diseases actually do. And I feel like awareness does help us, for example, us scientists to do public outreach. And it also helps people to be more aware and skeptical about, what do these pathogens do to our body, immune response-wise, for example. And I guess that's a positive thing that the pandemic has done to malaria research.
Yeah, and I think from my perspective, working on the clinical trials, the average person that comes in now has a much greater understanding of what we do and how everything works compared to 5 years ago.
Good. Nice to hear something good about it then. The next question comes from Dave Jenkins, I'm guessing. Do you have any thoughts on the other approaches to eliminating malaria, such as mosquito modification?
Oh, I love this question. I like mosquitoes. I mean, it's a love and hate relationship. They're nasty insects, but they're also fascinating. So there has been approaches where they try to genetically modify mosquitoes. I'm personally not sure if they've done this on Anopheles mosquito, which is the mosquito that spreads malaria. But I am sure that they have done this on Aedes aegypti, which is the mosquito that spreads dengue fever, the yellow fever, the West Nile virus, chikungunya, all sorts of nasty arboviruses. So they have done some genetic modifications in which they try to make the offspring sterile, for example, and they've done this in countries, for example, in Singapore. And I think it's a really cool approach. And there's also in Indonesia, where I'm from, they've been trying to utilize this naturally occurring bacteria that lives inside the mosquito called Wolbachia. And these I am not exactly sure about the mechanisms, but I am sure that these are strategies that have been used or trialed at least to control the mosquito population. But yeah, there are definitely genetic modification approaches in a way where we can tackle malaria.
Right. And these are being actively pursued as well, are they, in other places?
I think so. Yeah.
Yeah,
Right.
but I am quite skeptical of those, because I think it's always good to keep in mind that we should be using multiple approaches to kind of tackle a complex disease like malaria, and relying on something like just vector control, I think can render us vulnerable in case that ever fails, or perhaps a subpopulation of vectors where this doesn't work suddenly submerges. I'm not quite sure even how effective it is controlling that in the wild. That's not really my expertise, but I think what quite fascinated me when I read about this recently is that there have been studies that showed that those areas where vector control was quite successfully pursued, so meaning getting rid of mosquitoes, lots of bed nets, making sure that people don't get infected at all, those places tend to have like epidemics every now and then where malaria kind of breaks through, and because people haven't been exposed for a while, the natural immunity weans off, and then people tend to have gotten more sick. So there's some interesting epidemiological research on that as well. So if I were to support something, it would be something that, us as people, our bodies are able to deal with malaria if it comes to us, and less so messing with the environment or with the vectors that might transmit it. That's my opinion.
I do apologise for keep jumping in and asking my own questions, but you keep raising them. And that is, you're talking about the infections being delayed. Is it different in very young children and adults? Is it more severe in older or younger people?
There are differences in people's ages, but what seems to matter more is whether or not it's your first infection in life or not. So in areas that are quite heavily affected by malaria, it is mostly children who are vulnerable because that's usually when people get their first infections. However, there have been studies done with people who get their first infections later in life. For instance, people that live in Europe, for instance, and then go to areas to travel, catch malaria, and come back to Europe. And in those types of people, it does seem that the older you are, actually the more heavy your first malaria infection even becomes. And similar results have been shown in studies where people moved, like transgenerational migration, where large groups of people moved from one place to the other, and the new home had malaria and the previous home didn't. And then suddenly you could also see that the older people in the community suffered more. So delaying the first infection might not necessarily be a good idea. But yeah, people are working very hard in our field now to kind of piece all these informations together and better understand actually what's going on. And we hope that our work, which is, very cellular, trying to understand the mechanisms on a cellular level, hopefully can contribute to those answers.
Do you think, David wants to know whether you think that malaria can ever be eradicated, or will it always be endemic?
I mean, that is an excellent question that I think we would all like to know the answer to. I certainly think that, malaria is a disease of poverty. We've seen countries have been successful in eradicating malaria, so we know it can be done. And it's always going to be through a variety of strategies. We're not trying to say that malaria vaccines are the answer to the question here. I mean, vector control has been hugely effective historically. So I would say I am cautiously hopeful that it can be eradicated. The thing that I am nervous about is climate change and the risk of reintroducing malaria infection to places where it has been eradicated, and how are we going to get around that?
Just a small fun fact. It's not really a fun fact. It's kind of a scary fact. So there's this study that found out that mosquitoes in the sub-Saharan Africa region of the Salkeld region in Mali, the mosquitoes can actually fly up really high and ride the air current so that they can somehow repopulate areas that somehow has no malaria. And that's why these areas somehow see a lot of residual cases that they can't seem to get rid of. So that's something a bit interesting that people might want to know more about.
This is why you like the mosquitoes, isn't it? Because they're clever.
Yeah, and they're nasty buggers. They are.
Right. There's a follow-up question to the climate change one comment that Joe made actually here. It's from an anonymous person who commented on the ague, which used to be prevalent in the Fens and in other marshy areas of England in the past, and wonders whether you think it may return and we'll have to use your vaccines locally.
So yes, there was malaria in the UK and England historically. The last sort of local cases occurred in the 1940s, so it's not centuries ago, it's reasonably recent. As to whether we'll need to use our vaccines, so again, yes, climate change is a concern. Will we get reintroduction of mosquitoes that are capable of transmitting malaria? Possibly. So in the US, they've had their first transmissions of malaria via mosquitoes in the US with people without any travel history in the last year. And they hadn't had that for 30 years before. So obviously that is rather worrying. Historically, the malaria that was in the UK wasn't falciparum malaria, which is what our vaccines are targeting. So that brings up another point that there are different types of malaria, and we don't just need vaccines against falciparum, we need vaccines against other types. And we are working on vaccine for one of the other types, Vivax in Oxford. So there is ongoing research there.
And I think thinking back still with the climate change idea, Nadia mentions that the map shows an absence of malaria in Russia and China. And she says, is that because of the climate, that it's colder and drier there, or is it because we just don't know?
Might have to answer that for a little bit. So I'm not sure if there's still currently malaria in China before, but I'm sure that there was malaria, at least in the southern part of China, where it's warmer. For example, the island of Hainan, which is pretty near to Southeast Asia. And I think there was also a small outbreak of Plasmodium vivax malaria in Russia, Moscow, in the early 2000s. So I think they do exist. I am not exactly sure why they're not represented there in the world map. But yeah, they did for sure exist in those two countries.
Perhaps it's also worth noting that both of those countries have like vast areas that are not very densely populated, and I can imagine getting epidemiological data from those areas might be difficult as well. So it might also be that influencing what we have on those countries, or political reasons. I'm not quite sure either. Just my guesses.
So I just had a little look, say for China, and the good thing I think with malaria eradication is that countries are being declared malaria-free all the time. So China was actually declared malaria-free in 2021.
Good. Let's hope long may it stay that way.
Yes. It's good news.
Sticking with Nadia for questions, she wonders whether you envision any political factors that might negatively affect the large-scale deployment of a new vaccine.
I think it's a difficult question. I'm sure there's a lot of interesting anthropological and political science that can be done on malaria as a whole. But I can imagine that perhaps cost might be a factor on rolling out these vaccines, and whether or not governments want to prioritize that.
So continuing on what Katharina said, I think it's also the willingness to to invest in infrastructure, because at the end of the day, there's a lot of logistics involved in distributing the vaccines to be able to get them to remote areas. So there might be some political influences in that. Like, for example, the voted party, are they willing to invest a lot in infrastructure for healthcare to be able to roll out these vaccines to remote villages?
And the other thing is, I think, like, vaccine skepticism, vaccine hesitancy is not just a Western problem. It's something that occurs worldwide and something that needs to be addressed as part of the rollout of malaria vaccines.
And another interesting factor I think that's worth mentioning is that, like mentioned before by one of us, it is a disease that affects mostly poorer people and often is kind of the worst in rural areas as well. So whether governments want to prioritize into investing into that might be another question. So people who are wealthier might have better access to healthcare, meaning they might be able to get anti-malarials quicker if they or their children are infected, whereas somebody who's poorer or somebody who lives rurally might not have the same access and might therefore benefit much, much more from a vaccine.
In a related question, Dave Jenkins wonders how viable the vaccines are in terms of cost for large-scale rollout. And again, he refers, like you did, to the poor areas where they're most needed. Do you know much about the how impactful cost might be?
Well, yeah, cost, I mean, is hugely important. So RTSS, the first vaccine that has been rolled out, is significantly more expensive than R21. So we are making progress towards a cheaper vaccine. And yeah, the question as well is who's going to fund the vaccines? So you've got people like Gavi, Vaccine Alliance, that funds a lot of the vaccination programs in these poor countries. Will they take on funding for malaria vaccines? There's also malaria control programs. Will they fund them? So there's a lot of questions still to be answered in terms of how we're going to afford these vaccines.
Moving on to the next question, which is a big change of approach, change of question. Kat H asks, is there a benefit to blood stage vaccines against pre-erythrocyte vaccines? Is there any benefit to blood stage? Are blood stage vaccines better than pre-erythrocyte vaccines?
That's a good question. I don't know whether they're better. There are certainly advantages. One of the problems with these pre-erythrocytic early stage vaccines is that it's trying to block the parasite getting into the liver. If it fails to do that, if the parasite's released into the bloodstream, you get no protection from the vaccine. You might as well not have had one. With the blood stage vaccine, it's trying to sort of mimic more that natural immunity, that tolerance of the parasites in the blood without making you sick. So it's not an all or nothing, does the vaccine work or not question. There's also a concern with the pre-erythrocytic vaccines that whilst the children are getting vaccinated, they're not building up their natural immunity to malaria because they're not being exposed to the parasites in the blood. So what happens when you stop giving the boosters? Are they suddenly going to have rebound infections and get really sick with malaria? We don't really know the answer to that question yet. And again, that's something that blood stage vaccines can avoid. I mean, ultimately, what we're probably going to end up with is these combination vaccines. So targeting more than one stage and getting the best bits from both vaccines.
So you're saying the blood stage vaccines will be like the COVID vaccines have been here, that they stop us getting very ill and we're building up our own immunity, but we still do get a bit ill.
Well, yeah. I mean, hopefully you're not getting ill, but you might still have parasites in the blood.
Mm.
Yes.
Oh, right.
I think that's a good analogy.
Our research is also quite invested in this topic as well, and we hope that by being able to understand underlyingly what happens exactly with the immune cells, we can hopefully inform these questions and decisions as well. So if our research really supports what Joe was saying there about the blood stage vaccine, and that we can show with our research that it's particularly effective and safe, and can protect you also between different strains if you are able to build up your natural immunity while also having the vaccine and being protected from this severe disease, then that can influence the debate. So there's lots of work still ongoing, and hopefully with this upcoming trial we can help and influence that debate and hopefully come to some useful conclusions.
I'm thinking about different kinds of vaccines. Dave Jenkins asks again, do these have to be preventative vaccines, or can they be used after you've just been infected?
So, certainly what we've been working on have been preventative vaccines. And the other vaccines that have been licensed are also preventative vaccines.
And also, as we've discussed, your first infection or your first few infections might be the most dangerous. So you hope to kind of get a protection before you get to that point. But if you've had an infection, it might inform your natural immunity, but you might still benefit perhaps from getting a vaccine shot to be safer the next time you might catch malaria.
Yes. So you can still have the vaccine after you've had malaria, but it won't stop you getting sick in a particular malaria infection if you give it at that point.
Right. And Kat H comes back with another question about the dangers our immune response can cause to us. Because she asks, is it possible that the immune response gets milder with each infection, since it can be the immune response that kills?
That's a really great question. So that is really relevant to what we do in research in our lab. And with a previous challenge trial that we did, we actually found out that sort of, because when we actually challenge these participants for the first time, we study their immune response, we see this explosive response of this type of immune cell called T cells. It comes to the point where the T cells actually damage our own tissues and cells, which we don't actually want. But after the second and third infections, we actually see this excessive response being silenced. So we don't see this redundant response anymore. And we don't see this cytotoxic or cell-damaging types of T cells anymore. So they kind of become milder, or in a way more refined. We don't need to activate all these redundant types of T cells that we don't actually need and are actually bad for our body.
So it's making our immune response more targeted, basically.
Yeah.
I've got another change of direction here, with a question from Gray, and they ask, could we ever make mosquitoes immune from catching or transmitting malaria? And also, could we release a benign strain of malaria that outcompetes the deadly strains?
That's an interesting question. So for the second question, could we release a benign strain of malaria that outcompetes the deadly strains? I'm not sure if anybody's actually working on that, but there are strains of malaria that are more deadly than others. So Plasmodium falciparum being the more deadly one as in comparison to Plasmodium vivax, for instance. And vivax seems to be more prevalent in some areas, and Plasmodium in others. And that certainly is linked to the death toll in either area. However, I'm not quite sure what determines which strain becomes more prominent in which area. Do you guys have opinions on that?
Well,
I think, sorry.
No, go ahead, Joe.
I was just going to say, in regards to making mosquitoes immune from catching and transmitting malaria. So obviously we can't vaccinate mosquitoes, but some of the malaria vaccines that I briefly mentioned are what we call transmission blocking vaccines. So they aim to make a human gets vaccinated, they produce an immune response that is then able to block the transmission of the malaria parasite within the mosquito. So we can certainly approach it from that angle.
And again, thinking about the different types of vaccine, Kat asks whether failed pre-erythrocytic vaccines have any benefit, for instance, in reducing the parasitic load.
I mean, yeah, that's a good question. Are we just saying, oh, this person still got malaria, therefore the vaccine failed, but actually they are suppressing that parasite count? And I think that's something that we don't necessarily know the answer to at the moment. It is one of the things that we are very interested in looking at in our current trials, understanding more about how it does work in terms of suppressing the parasite count. And then, yeah, I mean, maybe is it something we want to look back at other vaccines that we've written off? I don't know.
We're back to Igor, who's asking again, does the stuff you're developing have some exciting new technology that can help against other diseases?
So from our perspective, since we're immunologists, we're more interested in the side of the human body and how the human body responds to all of this. So we're not really working on technologies ourselves, but perhaps it can have a bit of an impact on how our research could have an impact how we approach other infectious diseases that are unrelated. So it's quite interesting how different infectious diseases that seem like unrelated to one another can really inform the research of the other. So in that sense, there's always, everyone benefits from more research.
Yeah, who knows where something's going to help until you do it. We've come on to our last question now, and this again is from Igor, who's wondering whether with this malaria vaccine you ever encounter any anti-science pushback. I'm sure it must have happened with COVID, but has any grifter accused you of spreading autism yet?
Yeah, that's a good question. And I mean, I think I'm working on clinical trials, but in my role, I'm very much with people who have volunteered to be there, want to be there, interested in vaccine research. So we don't tend to get that pushback, which is obviously very nice for me. So the Oxford Vaccine Centre is very active on social media, and they do get pushback. But yes, it's something that I'm in a sort of protected position for.
Good. Good. Well, thank you very much. That's actually all we have time for. I wonder whether it's possible to flash up that last slide with the QR code, because if anyone is available and willing to be made ill voluntarily, and I'm really sorry, you have to be under 45, otherwise I'd be right in there, I promise. So any volunteers, I think they would all be very grateful for. We are going to be opening the Lockins Razor very shortly, so we'd like to see you head on over there. And we will see you next on the 27th of June. So one last thank you very much for Joe, Matteo and Katharina. And yeah, thank you very much from me as well.
Thank you.